Hi, my name is Jessica Redline, and I am going to be talking about our Lab 3 results. This is going to be the video on the results for the agar plates for LAPU Micro 225 for Lab 3. So I retrieved my plates that were incubating in my cabinet above my microwave. So this is the dilution plate counts for Lab 3, and I was pretty surprised with the growth of the plates, and then seeing the whole process actually take effect of how with each dilution through each tube and after being plated, how each plate decreased in the number of counts. So with count plate number 1, you can see the growth. I was unable to count it because the growth was just so, there's one huge surface area that's covered by the colonies, and then there's some like sporadically over here, but there's just so many bunched together in this area, so that made it too numerous to count. On the plate 2, these ones had quite a bit and multiple in this area too, where it looks different than these specks, which was considered viable. I counted approximately 50. There's also another spot here that contains a lot of spots around it, which I counted. Plate number 3 had approximately 10 colonies, and these were bigger. So in plate 2, there was like different characteristics of the colonies. There were some like yellow, almost raised specks that were some big, some small, some smaller. And then there was almost like this fuzzy looking patch with like a bunch of specks around it. So I counted those colonies. So for the plate 3, I counted about 10 colonies. For plate number 4, I counted 8 colonies. This one's a little bit harder to see, but there are some small specks on the outside rims of plate 4. For plate 5, I got 6 colonies. You can see them there. And then for plate 6, I got 2, and they're super teeny tiny on the outskirts of it, but this, for the most part, this plate looks pretty much clear. And it shows that with each plate and each dilution process, it just kept getting more diluted each and every tube as I passed through each one and then plated it on the agar. So I thought that was interesting to see how the colonies reacted to the dilution process, and how the colonies became decreased with each plate. So I had a little bit of trouble with doing the equation for the CFUs count, but I just kept researching, examples of equations and, looking at the reading, and I think I somewhat got it figured out, but it's something that I definitely still have to work on and that I want to learn. But overall, I think this was a really good experiment, and it was a fun experiment to learn about how much the dilution of certain particles can affect the outcome of the sample. So thank you so much for watching for this week's Lab 3.